So in today's presentation, we're going to look at the process of meiosis and compare that to mitosis in some respects. So we'll look at some similarities and differences. So just to revise the process of mitosis a little bit, so mitosis, so what its purpose is and where it occurs and all those sorts of things. So the purpose of mitosis is for growth, repair, so you cut yourself, maintenance, so the continual replacement of cells that are worn out, blood cells, cells in the stomach, cells in the intestines, and asexual reproduction. So reproduction that occurs in organisms where they can reproduce by themselves. They don't need a male and a female for reproduction to occur. So growth, repair, maintenance, and asexual reproduction. Now, mitosis occurs in nearly every cell in the body except for the gonads, and we'll talk about that role in meiosis. So they occur in every single cell, and we call them somatic cells. Okay, so normal body cells. So an example of a somatic cell might be epithelial cells, skin cells. Okay. Now, the other thing we need to look at is just before we get looking at meiosis in any greater detail is just to look at this idea of some terminology of what chromosomes are. So in mitosis and meiosis, before cell division occurs, we look at the cells as being diploid. So diploid, which means the same has two sets of chromosomes. So two sets of chromosomes. Okay, or we can look at that in as 2 times n. So that was 1 chromosome, it would equal 2. In humans, it's 2 times 23, so it would equal 46. Okay, so 2 times n in humans would be 46. Just look at a little bit detail of how those chromosomes look. So characteristic chromosome might look like an X shape, that's what you remember. However, a chromosome is a piece of genetic material that has genes on it. Okay, and colour this one in pink because this has a maternal origin. Maternal origin, which means that it comes from the mother. And green has a paternal origin. Okay, so maternal and paternal origin. So that means that for every organism that is the offspring of sexual reproduction, there is a paternal chromosome and a maternal chromosome for chromosome 1, chromosome 2, and how many chromosomes that exist. Now, just before replication occurs, these chromosomes duplicate. And this is when we start to see this characteristic X structure. So we'll just draw this X structure in. Okay, and we call these things sister chromatids. So these are called sister chromatids. Okay, so sister chromatids of the paternal chromosome, and they are joined together in the center by what we call a centromere. So centromere. Okay, now finally the last bit of terminology is that our maternal and our paternal chromosomes, say for chromosome 1 in a human, is what we call a homologous chromosome. Okay, what that means is that on chromosomes there are genes, and there's areas that don't code for genes, and we're still currently working out what that's all about. And say for this particular chromosome in that particular location, the same gene would be there. So it could be chromosome 1, and that would be the gene for, say, hair color. So same location, same chromosome, and we call these homologous chromosomes. Now the process of meiosis, is this for a slightly different purpose, and this is for sexual reproduction. And saying that, when sexual reproduction, it is to produce gametes. So it produces gametes. Okay, so in animals, those gametes would be sperm for the male and egg for the female. In plants, the anther produces pollen, and it's an egg as well. Okay, and that's situated in the ovule. Okay, so it produces gametes, and it occurs only in the gonads. Okay, so in animals again, that is the testes and ovaries, ova, singular, and in plants, it's the anther, which produces the pollen, and the ovule. Now the ovule is what will produce the seed. So the seed within inside the fruit is an ovule. So we're going to take a closer look at this process of meiosis in a little bit more detail. So, as in mitosis, the first, or not even the first stage, before cell division occurs, we have interphase. Now interphase, this is where the normal cellular activity is occurring. So we have our cell membrane. Inside the cell membrane we have the cytoplasm, and we have our nuclear membrane. Towards the end of interphase, we see the chromosomes replicate. However, the material in there at this point in time is called chromatin. Okay, so it is all elongated and spiraling. It hasn't taken on that characteristic shape of chromosomes that we're used to seeing. So the first stage of actual cell division is prophase. Okay, so prophase. Okay, so in prophase, we start to see the nuclear membrane is broken down. So at this point, the process of meiosis occurs inside the cytoplasm. I should point out too, a little oversight from my point, this is prophase 1, and this will become clear why we call this prophase 1 in a moment. Now in prophase 1, we start to see that, as I said, nuclear membrane breaks down, chromosomes are split into chromosomes, and a process called crossing over occurs. So we're just going to draw that in. We're going to use two sets of homologous chromosomes to indicate this. Okay, so we have our first set, and we'll just make them a little bit smaller in terms of size difference. So our first set of chromosomes, and we'll do a second one, draw the maternal chromosomes in, and then we will draw our paternal chromosomes. So in this stage we see, as I said, crossing over occurring. Now what crossing over is, and we'll write that down there, crossing over, or chiasma, is we see some genetic material from one of the arms of the paternal chromosome crossing over with one of the arms of the maternal chromosome. So it's still chromosome, say chromosome 1, And I haven't drawn it in here, but it can occur at different points on all the chromosomes. It is irregular in the way that it does that and random. So we see crossing over occurring at this very stage here. So the next stage is metaphase. Metaphase 1. Okay, so we'll draw our cell membrane in, and we will draw our chromosomes in as well. So what we find occurring in this stage is that just like in mitosis, we see that they start to align in the center. So we call this line in the middle, or the equator of the cell. So we'll draw our chromosomes in. We'll draw our green one. And what I've forgotten to put in here is that there is a little, and I'll just draw over it a little bit here, there has been a bit of crossing over occurring, a bit of exchange of genetic material. Now the importance of this exchange of genetic material is that it increases variation. The variation is obviously an important thing, so it increases variation via crossing over. Now what we're occurring is that they're lining up at the equator, and I've placed the paternal chromosome 2, the smaller one there, on the opposite side of the equator to our chromosome 1, and same with our maternal chromosomes. So there's some crossing over causing variation which occurred in prophase, and they line up at the equator. Now at the same time as they are lining up, we see spindle fibers. Now these spindle fibers come from what we call a centriole, and the centriole extends from both sides. Okay, so a centriole. And they're little cylinder-shaped objects. Now they're very important because they actually they attach to the centrioles that are, sorry, the centromeres. Getting my terminology messed up. Centromeres, they attach to the centromeres, which join our chromosome, our chromatids together, and they later on will pull them apart. So the next phase is anaphase 1. Okay, so anaphase 1. So in anaphase 1, what we're going to see is that these chromosomes have been drawn away from the equator. So chromosomes have been drawn away from the equator. I'll have to put a little bit of green one on that later. Okay, we'll place our paternal one in. We'll just put our little glow on that. By these microtubules from our centrioles. They've been drawn away. Okay, and just finally put our little bit on there. Okay, so they've gone to either side. This is really, really important, and what this does is also increases variation, because as we can see, the two chromosomes have gone to opposite poles in terms of the paternal and maternal, and this is what we call segregation or independent assortment. The independent assortment. Okay, and that's a really, really important process. Not only is chromosome 1 going to either end before the first part of the meiosis occurs, the first cell division occurs, but also that they're going to different locations. So the resulting egg or sperm are going to have a combination of maternal and paternal genes. Following anaphase 1, we move on to telophase 1. Telophase 1. Okay, so just like in mitosis, in this part we see it's really, really small. So I might just erase that out and try and do that a little bit bigger. Not too big, just a little bit bigger. And I don't think that's much better. A couple of things start to occur. We have our chromosomes forming, again with that variation. Crossing over occurring, and we're starting to see the very, very beginnings of cytokinesis occurring. Okay, so also what we see is the chromosome numbers are halved. Each daughter cell here, and the two daughter cells have half the number of chromosomes. Finally, the last phase is telekinesis 1. Cytokinesis. So the last stage is cytokinesis 1. So in this stage, we see the formation of two daughter cells. They're pinched off here, and obviously this has been an animal cell, and our resulting cells will have half the chromosomes that they had before. The other thing to note that these cells they're no longer identical. Okay, so we've got some exchange of genetic material for a couple of things, some variation from crossing over, some variation from this process of independent assortment. So these are not identical. After cytokinesis 1, we move into metaphase 2. So this is the second cell division that occurs in meiosis. So meiosis occurs in 2 cell divisions. So we'll draw our 2 cells in. Of metaphase 2. So straight into metaphase 2. So in metaphase 2, our chromosomes are going to align again at the equator. So we draw our chromosome in. This is the one that's got a little bit of variation in it. Place the green arm on it. And we'll draw our smaller chromosome 2, and we can see that they're aligning at the equator, and the same for the other one. This is the one that's got a bit of variation in it. A little mixed up from before, but we can see that it still retains that variation. Okay, so they're aligning at the equator. Again, microtubules will form. This time in this orientation, coming from our centrioles. So centriole. Okay, so microtubules forming on both of these. After metaphase 2, we move into anaphase 2. So we can see the naming, the terminology is similar, and what's occurring in each stage is similar. So in anaphase 2, I'll draw in my two cells, similar size, and we will place in our chromosomes. And what's occurring in this stage is that they're actually starting to be drawn again to opposite ends of the chromosome. Okay, so this is the one that's going to have the slight variation that occurred from crossing over. So we'll draw that variation in, not all of it. Variation in it. And we draw our smaller one. Okay. And again, we'll do on our other cell, we can see again they've been drawn to either end of the cell. And we'll put our variation in on that one as well, from the crossing over, and our smaller cells. So they've been drawn by microtubules to either end of the cell away from the equator. So chromatids move away from equator to either pole. Okay, so the final stage is cytokinesis 2. I'm just going to place cytokinesis 2 over here, and it's going to require so 4 cells being made. Okay, so in cytokinesis 2, we see that each cell reduces in number one more time. So after anaphase, we see the cells reducing in number one more time, so that we have and we'll put in the green. And a little bit of pink. Okay, so we've produced our four daughter cells, and it's important again to note that they are not identical to each other. So 4 daughter cells, and they're not identical. Okay, so and we can see that variation occurring there, and also that each one has a slightly different variation, whether that it has the paternal or the maternal gene for chromosome 2, or vice versa for chromosome 1, the maternal or paternal. So we can see that variation. Now, depending on in fertilization, if this was the sperm, depending on which sperm actually undergoes fertilization with the egg, it very much dependent on what material will be passed on. So these are our possible scenarios, but it really depends on which sperm actually causes or is able to fertilize the egg to understand what genes will be passed on in that particular event. Now, the other thing to recognize here, that the daughter cells have half the number of chromosomes. So daughter cells have half the number of chromosomes, and we term this haploid. So diploid is 2n, or 2 sets of chromosomes, 2 sets of each chromosome. Haploid is just n. So it's 1 set of each chromosome, and it produces 4 daughter cells. Okay, with haploid number 1 set of each chromosome. Now, it's not until this process of fertilization occurs So in the process of fertilization, we see that the haploid number will again become diploid number, so having 2 sets of chromosomes. And that occurs because our sperm, we'll put our little tail on the sperm, and our egg both contain half the number of genetic material. They contain half the number of genetic material. Okay, so we'll place the sperm again being in green. So we have our 2 chromosomes, both of paternal origin in this case, and the mother will have 2 chromosomes, both of maternal origin, and when they join together, we finally get our cells having diploid number again. So when they join together, So through fertilization, joining of our egg and our sperm together, we produce a new cell, and in that cell it will have maternal chromosomes and paternal chromosomes. So again, we can see that this diploid number has been restored.